Hello everyone, my name is Deja Tillman. Today is March 29th, 2020. I'm doing my experiment a little bit early because I do have a busy schedule ahead of me next week. This is part 1 of our lab exercise, biochemical testing for microbe identification, methyl red, vulgar, and catalyst testing. So I'm going to be starting exercise 1 of the lab exercise. Before putting my cultures into incubation for 48 hours, I've already cleaned my area. I have everything set up. I don't know if you guys can see. I have my 2 broth cultures that we've had since week 1. I also have the MRVP broth cultures. I have labeled them E. coli and S. epidermis. So those are labeled, and they've been sitting out for a while now, so they're at room temperature. I have my alcohol here. My pipettes have alcohol sitting in them. And I have my candle lit. So I am ready to sterilize my tubes. I have my gloves on, my apron, covered toes, my goggles, and my mask. I cleaned my surface with bleach already. And now that everything's ready, I'm going to go ahead and sterilize my cultures here. So take the top off, run it over the candle. So here's the E. coli culture. And here's the MRVP broth, labeled E. coli. I'm going to sterilize the tip of this as well. You're just going to spin it. And then I'm going to set this here so you guys can see. It says then to remove one of the droplets with alcohol and shake it dry. Now, we're going to insert the tip of the pipette into the active E. coli culture and get a small amount. And I'm going to then transfer it over to our MRVP labeled E. coli. So I transferred that. This is going to be dumped in our bleach solution over here in this corner. And I'm then going to go ahead and sterilize the tip of the MRVP. Then I'm going to seal it closed. So this tube will go into incubation for 3 days. I'll leave that to the side. That one is ready. I'm going to sterilize the E. coli that's been sitting for weeks. And you see my culture is still actively present in there, the E. coli. And this is just going to save. Now I'm going to repeat the same steps for the S. epidermis. So I'm going to sterilize our culture that has been sitting. This is the MRVP broth. And I'll be putting the active culture in this MRVP tube and also let it sit. Okay, so both sterilized. Then I have a new pipette with alcohol. I'll squeeze the alcohol out. Make sure it's dry. Then I will get a sample of the S. epidermidis. And then put it in the MRVP. 1, 2, 3, 4. About 4 drops here. And then putting into my bleach solution, we're going to sterilize the top. Both is going to go into incubation for 2 days. And S. epidermidis will keep. And this would be the end of part 1 video. I will be checking back on these broth cultures in 2 days, 48 hours, and we will see what grows. We'll do our methyl red and Vogus Prosker test and the catalyst test after it sits for 2 days. That'll be part 2. Part 1 is complete. Thank you, guys. Alrighty, guys, I'm back. It's still the same day. It is March, I keep forgetting the date, 29th. My name is Deja Tillman, and I'm gonna be showing you, I have my E. coli slide and my S. epidermidis slide with the dime-sized circles. And for both these slides, I'm gonna be scraping the culture that are in these plates, and I will be adding a drop of hydrogen peroxide to each slide with the culture on it. We're going to see exactly what happens, if it bubbles up or not. So I have my inoculation loops and alcohol, and I'm going to start with the E. coli. And I'm going to scrape some cultures, make sure the loop is dry. Scrape some cultures off of here. And I'm going to put it in the dime size area of the E. coli here. Perfect. You see it there. I'm going to drop this into the bleach solution. And I'm going to grab the S. epidermidis next. S. epidermidis. And then also place that into the dime size culture on this one. That's the S. epidermidis. And then 1 drop of hydrogen peroxide is going to go into the sample. So I'll get a clean cup here, put some hydrogen peroxide, and then I'm going to put some into the pipette here of hydrogen peroxide. Here you can see. I'm going to grab a little bit of it here, and then we're going to see if it bubbles. So our S. epidermis, add 1 drop. And I definitely see bubbling. I am going to take a picture. And I see some bubbling on the S. epidermidis, a little bit more than the E. coli. So I'm going to show you guys here, if you can see. This one, S. epidermis, is bubbling way more than the E. coli, but the E. coli is also bubbling. I am going to take a picture right now, and I'm going to end this video. And that will be the end of this catalyst testing. Thank you.